So, welcome to the topic of cell and its subcellular organelles, and I will be discussing about it in brief and in tabular column. Let me begin with the table of contents, or what we will be discussing here is the term or name of the subcellular organelle along with its measurement and synonym, about the subcellular organelle and its function. So let me begin with actin filament. We can discuss about it in microfilament. Next comes the basal body. It is about 150 to 500 nanometer in diameter and in height, and 250 nanometer in diameter. It is known as blepharoplast or kinetosome. So wherever these two names are coming, the author wants to talk about the basal body. It is found at the base of the cilia or flagella, and it is formed from centriole. So some subcellular organelles are connected to each other. It helps in the functions as motility, sensory function, cell cycle progression, morphogenesis, and sensation from the environment. Moving on to the next subcellular organelles and cell itself. So cell is a basic unit of life. We all know that when it is grouped together, it forms tissues. Tissue, it is a level of organization where structurally similar and functionally similar cells are grouped together to perform the function. It is 10 to 20 millimeter in diameter. So the functions of cell are absorption, digestion, respiration, excretion and egestion of the nutrients along with its secretion, biosynthesis, that is formation of nutrients, movement from one place to another place, irritability to sense and respond to changes in its surrounding, and homeostasis is maintenance of the inside and outside of the cell environment. Reproduction, as we all know that mitosis and meiosis will be taking place in the cell. Next is centriole. It is 250 nanometer in diameter and approx 500 nanometer long in vertebrate cells. So it is a minute cylindrical organelle and it is occurring in pair. It is always occurring in pair and formed of 9 group of microtubules triplet organization. Centrioles transform into basal body. Just now we have learned or discussed it in the section of basal body, and it helps in cell division. Basal body which is present at the base of the cilia. Next is centromere. Centromere or kinetochore, it is the region of chromosome to which sister chromatids attached. Regional centromeres are a size of 100 kb to 500 MB. It helps the DNA during cell division, DNA division. From this point only DNA will get divided. So it is helping the DNA division during cell division. Next subcellular organelle is centrosome. Let me clear this. Centrosome. Centrosome average length is 0.3 to 0.5 μm and diameter is 0.2 μm. It is known as its synonym is microtubule organizing center. It contains centrioles. Centrosome contains centrioles which are lying perpendicular to each other, from which spindle fiber develops later on. So it is a regulator of cell cycle progression and maintain chromosome number during division. As we all know that there will be a 46 pair of chromosome in human being or Homo sapiens, 23 pairs that comes from male gamete and 23 pairs that comes from female gametes. So this division or maintenance of 23 pairs from one male gamete, 23 pairs from female gamete, another gamete. So this division will be maintained with the help of this centrosome. Moving on to the next subcellular organelles, they are, that is chromatids. Chromatid, its diameter is 765 nanometer. So it is also known as chromosome 2 halves. So one of the two identical halves of a chromosome. So the coiled part is chromosome. So when this chromosome when we will be seeing in detail that identical two halves. Identical two half, this single part is chromatid. So division and distribution of chromosomes to a new daughter cells. So this chromatid will go to one cell, this chromatid will go to another cell. I mean two different daughter cells. They will get divided from this point. Next is chromosome. Largest chromosome 1 is 250 million nucleotides long, whereas smallest chromosome 21 is 40 million nucleotides in length. So largest chromosome is 1, smallest chromosome is 21. So it is a genetic material of the Homo sapien or the organism. It is a thread-like structure, consists of protein and DNA. Chromosomes consist of protein and DNA and allow DNA to be accurately copied during cell division. With the help of this chromosome, the DNA will get accurately copied during cell division from one cell to the two different daughter cell. Next is cilia. So cilia, it's is arising from basal body, and this basal body it is formed from centriole. So length is 1 millimeter. This length is 1 millimeter in chondrocytes and up to 30 millimeter in kidney epithelial cells. So largest cilia found in kidney epithelial cells. So it's an hair-like structure, whereas some differences are there in cilia and flagella. We'll be referring in primitive organisms section. So it is primitive in nature. So it is found from the low level of organisms to the highest level of organisms, and found it is found either a single or many. So this cilia they will be found either single or in tufts. So movement of cellular fluid, it helps in movement of cellular fluid in both direction. It whips back and forth so that cellular fluid move from here and there. Next is cytoplasm. Cytoplasm is 70% of the cell's volume is cytoplasm, leaving about the cell nucleus and biomolecules. So it is also known as protoplasm, cytosolic organelles that cytoplasm contains, and cytoplasmic inclusion. And it is in colorless media for cell organelles in which that cell organelles are submerged or they are floating in short. So it helps in maintenance of cell shape, protects from damage and material exchange inside to outside of the cell. Moving on to the next subcellular organelle, that is cytoskeleton. Cytoskeleton, that measurements are given individually, and it is also known as cellular skeleton. That cytoskeleton includes microfilaments, intermediate filaments, and microtubules. Microfilaments, just now at the initial of this document or initial of this session, we have seen it is also known as actin filament. Next, it helps in maintaining shape and internal organization. As we know that it is a cellular skeleton, so by it is giving in shape and internal organization of the cell, maintaining, and it also provides mechanical support. Diplosomes, centrioles. We will have to see in centrioles, which we have already checked. It is known as centrosphere or kinoplasm. Diplosome is known as centrosphere and kinoplasm. Don't get confused it with centrioles. Pair of centrioles arranged perpendicular to one another. So that centrioles, which are long cylindrical tubules, which are arranged perpendicular to each other. That arrangement is known as diplosomes. And this region is known as, just now we have studied. And so next is diplosome. So we will be seeing its measurement in centrioles. The centrioles are long tubules which are arranged perpendicular to each other. So that arrangement is known as diplosome. It is also known as centrosphere or kinoplasm. It helps in cilium movement and mitosis. Pair of centrioles arranged perpendicular to one another is known as diplosomes. Just now we have seen, let me go to the region, that is centrosome. Centrosome region, which contains centrioles in animal cells, is centrosome, and its length is 0.3 to 0.5 millimeter and diameter is 0.2 millimeter. Whereas diplosome is the centrioles which are arranged perpendicular to each other. So today we have completed some of the subcellular organelles. So in the next sessions, we will be seeing or learning about other subcellular organelles. Thank you.